Hi, everyone, and welcome back to The Science of Turning Back Time, the podcast where we unpack the latest science and strategies behind healthy aging and extending your health span. I want to start this episode with a personal story. When I first ran my true age epigenetic test on myself, I was quietly confident. I eat well, I move often, I prioritize sleep, I do stress reduction. So I was expecting to blow my chronological age out of the water. But the results, well, underwhelming. I was just 3 years younger than my actual age. And honestly, I felt a bit disappointed given this is my field of work. So that's the reason I started this podcast, and really for myself, but hoping to bring my listeners along with me. So the good news is I've since been able to bring that number down even further, which has been deeply rewarding and very motivating. But that's what today's episode is about: the science behind biological age, how we can measure and influence it. And I'm thrilled to be joined by someone who's right at the heart of this field, Dr. Varun Dwaraka, the head of bioinformatics at TruDiagnostics. So Varun's background is in tissue regeneration, and his current work is helping thousands of people track and transform their biological aging through DNA methylation data and epigenetic clocks. So, Varun, welcome and thank you so much for being here.
Well, thank you. Thank you so much, Charmaine. And it was actually really nice to hear your personal input and your take on the True Age test, which is kind of great because especially as developing a lot of these tools, it's really good to see how it's being used. So thank you for that.
Well, let's start with your journey. Can you share a little bit about your background in tissue regeneration and how that's led you to your current work in epigenetics and biological age?
Yeah, absolutely. I will say my path was a little bit non-traditional. Growing up, I've actually been in labs since my first year of, I guess, uni or university years. Um, and I've done everything from, understanding plant genetics to understanding cancer genetics to during my PhD, really focusing on tissue regeneration in salamanders, which is a surprising twist from what I study now, which are humans. Salamanders have this insane ability to be able to regenerate virtually all parts of their body. So their tail, half of their brain, entire limbs, spinal cord. And a lot of the questions that I was kind of studying during my PhD was what are the genetic and epigenetic mechanisms that really were allowing for this tissue regeneration to happen? And as I was going through finishing up my dissertation, one question just kept popping up is how are these cells that were lost able to understand the age at which they were, because not only are you regenerating the original appendage by the same exact form and function as it used to be, but they also know how to modulate that aging requirement, so how to get back to the same age. And so that led me into reading a lot more of these aging papers, and this concept of aging just started to kind of rattle in my head. So after I was finishing up my PhD, I actually wanted to go towards a purely academic, let's say, journey, if you will, after my PhD. But literally down the street, I did my PhD at University of Kentucky. Down the street in Lexington, Kentucky, was a small company at the time called TruDiagnostic, which was really utilizing DNA methylation, which I studied extensively during my PhD years, and using that as a diagnostic, and also for the purpose of understanding what your biological age was. And there's a name called Steve Horvath, which I'll maybe explain a little bit later, where a lot of Steve Horvath's work in DNA methylation actually helped a little bit with my PhD, but a lot of that technology and a lot of that of the science that Steve Horvath was kind of spearheading also was something that True Diagnostic was leveraging. And so it really worked out that, in almost like a series of fortunate events, I was able to transition over to TruDiagnostic as then the head of bioinformatics, and be able to contribute to developing not only the current state of the true age, but also developing newer tools and a lot more refined tools that are now being offered from TruDiagnostic. So, really, it was this concept of just trying to understand aging, understanding how epigenetics and genetics really related to that aging process, and then later on, how can we provide that back to the end user for really giving them an actionable output that they could use for their health outcomes.
It is very exciting. And some of the things we'll talk about today is how you test, because, yeah, I mean, let's actually start with that. How do you
Yeah,
test for biological aging? And, could you explain a little bit the difference between chronological and biological aging, and how do you test for it?
absolutely. Yeah. So chronological age is nothing but a fancy, terminology to say your age based on your date of birth. So this is assigned based on the date that you were born, and is essentially the calendar years of how long you've existed in this world. But the thing about aging is that aging can be defined as one of the definitions of aging is the exposure that you have with your environment and how that affects your form and function of your daily cellular and overall processes. And so, with that kind of definition, I think it's easy, maybe somewhat clear to see that sometimes just existing in your environment can have differences in how your body will respond. So, if you exist in a very harsh environment, your aging processes might be greater, and thus your form and function might be deteriorating at a faster rate. And so, this is the concept of separating out just the calendar years to your biological age, which is more how much of your biology has been impacted with your existence in the natural world. And so the way that we're testing biological age is actually using those cellular dynamics, so those epigenetic marks that are available in your body. I'll go through that definition in a little bit, but how those epigenetic marks are being translated into a biological age that is representative of your cellular age and also a proxy of your cellular health. So the way that we're testing it is that we're using blood. It's essentially a standard blood test, and we have the technology to capture your epigenetic changes. So when we talk about epigenetics, that's another thing that's very different from I think what a lot of listeners know about your genetics. So your genetics is going to be based on your DNA. Your epigenetics are the actual, in this case, the specific marks on your DNA, the DNA methylation marks that are able to modulate how your DNA is being interpreted. And so, what this really kind of boils down to is that even without changing the DNA itself, you can actually modulate how your DNA is being read, which can then facilitate different biological processes, or in the case of aging, certain biological aging processes to be increased or decreased, or be protected, or be opened up to be deleterious. That can facilitate how your age is being increased or decreased. And so, with that, we're really using these epigenetic marks from a blood test to quantify using all these different machine learning models that we've created and that the field has created to give you a value that you can say, are you increased or decreased from your chronological age?
So that is very exciting. And as you're saying, this is actually something that people can influence and they can change.
Yeah, and it really comes down back to the biology of the biomarker. Because one of the things that I think a lot of people maybe they know this, is that you can actually create a biological age based on so many things. For example, Peter Attia, I'm a fan of Peter Attia. He talks about how VO2 max and your functional scores are really the biggest driver of biological age. As much as I agree, those functional aspects are things that have already, come about. By then, the changes have already been made, but when we think about epigenetics, the thing about these epigenetic marks is that they're really heavily influenced by your environment, just as from a biological standpoint. In fact, actually, these epigenetic marks, the DNA methylation marks, are readily changed based on your exposures to, pollution, the diet that you have, the alcohol intake that you take in. Even things like stress actually influence your epigenetics to make those biological modifications later on. And so why not create a epigenetic age, or sorry, a biological age based on those modifiable epigenetic marks? And that was the whole thesis of being able to bring this type of test to the market.
So when I sent away my first test, I had expected to get back a result, like a one pager, which just says you are 25,
Yeah, yeah,
whatever, just with one number age on. But actually, I've got the test results here in front of me. I've got
mhm,
pages and pages and pages. And it's a lot more complicated than that.
yeah,
So there are lots of different things that you look at to sort of combine a whole picture. And can I ask you about some of these specifically?
sure, absolutely,
Uh, so yes, so like I said at the beginning, is that through this podcast and looking at aging, I've been working really hard to see if I can influence some of these numbers for myself. And so the gap between my tests was 7 months. And so my telomere length is one area which I have influenced quite a lot, quite significantly, and have brought down my biological age quite a bit through telomere length. Can you explain to the listeners what telomere lengths are, and being able to change that number in terms of telomere length? What does it mean for me that I've been able to increase the telomere length?
yeah, absolutely. So telomeres are essentially those protective caps at the end of your at the end of your DNA strands, which are essentially protecting the important parts of your DNA from being chewed up by the environment of your cells. And then actually, typically, we see increased shortening of those. So by the time that those telomeres are shortened, the reason why the shortening or the reduction of those telomere lengths are so important and deleterious to your health is that by the time you get very, very low in that telomere length, you can start to have the important DNA be impacted, which could then cause, deleterious effects, cancer being one of them, just those cancer states. And so for the longest time, telomeres were actually quantified using genetics and actually using something like PCR, which I think nowadays everyone knows what PCR or polymerase chain reaction is, where you could use PCR as a way to quantify the total length of that piece of that telomere. Our telomere test is actually a reimagination of that, using epigenetic marks as a way to estimate what your presumed telomere length is. And so by increasing your telomere length, or sorry, the reduction in telomere length could also be associated with increased cell cycle. So the more cell cycle divisions that you occur naturally in your body, you actually start to hit a finite amount of cell cycles. So it's almost like, let's say with the car, you have a 200,000 mile warranty in your car. So once you get closer to that 200,000 miles, your car starts to break down. It's the same with your cell cycles. And so telomeres are essentially an estimation of that. So what you could do is that by taking this test, the reason why those telomeres were increasing could potentially be that the epigenetic processes that control the regulation of the telomere of the telomere lengths, or the telomerase activity, if you will, the protein that actually controls how much telomere is there, um, could be increased through this process. So whatever you were doing, not knowing much about what a kind of interventions you were taking, you could actually revert that by either A, slowing down the number of cells, the cell cycles that you're going through, or B, increasing the telomerase activity innately through whatever intervention that you were doing. So essentially, telomere length is quantified by that, and that's why we want to see an increase in that telomere length, because that technically represents a younger cell type.
And Varun, so I wasn't doing anything like crazy or unusual. I decided to just really focus on um, very much cleaning up the diet, making sure there's no processed foods, increasing
Yeah,
exercise, making sure I'm sleeping well. Um, you know, this second phase, I'm going to be now experimenting with a few more interesting things. But this was really just the real basics. And so it's been very interesting to see how that's changed. Now my
yeah.
omic age has gone from 46 down to 39, which I
Wow.
was really
Yeah.
excited about. And again, I haven't done anything hugely dramatic, except being very consistent with kind of lifestyle things. What is omic age?
Yeah, so omic age, actually, that's one thing that we're proud of at TruDiagnostic, because that was actually a clock that we built in-house with our collaborators at Harvard. So omic age is this imagination of an epigenetic clock, but the thing about molecular biology is that epigenetics is one facet of the entire molecular biology that's available. So I'm sure many of your listeners understand proteins. They may understand metabolites. They may understand, gene expression and all that. And so while epigenetics is a very important portion of how your DNA is being read into the gene expression and being able to contribute to protein synthesis, we understand that aging is contributing, or is contributed, not only to your epigenetics, but to your protein information, your metabolite information, and also your general clinical information. Right? Like whenever you go to the doctor, you're taking a standard lab panel like your IL-6, CRP, your even your VO2 max. We wanted to figure out a way, how can we synthesize all of this information into a singular epigenetic clock? And that was what omic age is for us. Is it was a way to statistically take all of this information into one single clock and then be able to provide it to the end user. Now, the cool thing about this is that when you send in your blood, because I believe you took just a standard blood draw, right? It was just a dry blood spot card. We didn't ask you for any type of VO2 max information. We didn't ask for any of that information, right? The reason why that's possible is because through the usage of machine learning and AI, what we could do is that we could take the same DNA methylation data that we routinely extract to calculate all of this telomere length and all this other information, but we could use the different models to estimate what your presumptive IL-6, your presumptive metabolite levels, your presumptive protein levels, and then use that in a way to synthesize all that information into that omic age clock. So with all of that said, your omic age is essentially a significant representation of your entire molecular health synthesized into one single value, which is your omic age. And also, just as a maybe for the listeners out there, omic is actually, if you're part of the sciences, omics are supposed to be the technologies that are able to quantify the different states of your biology. So that's why we named it omic, is because we actually took all of this information from the different technologies to synthesize it together.
So just very quickly, I mean, how quickly can these markers change? So if you start putting in interventions, how quickly do they change?
Absolutely. Yeah. So a lot of what we've understood from these changes are actually through these randomized control trials. So we were part of a randomized control trial about a year ago, which was published in BMC Medicine. In that study, it took about 8 weeks to be able to revert not only just one epigenetic clock, but 12 different clocks. And many of those clocks included, for example, the systems age, or I think we call it symphony age in our report. So I think about 8 or so of those 12 clocks showed reductions. Dunedin pace, which is the pace of aging clock, showed a reduction. So 8 weeks was originally the quickest we've seen, and even that intervention was just a diet change. So I think earlier you were mentioning that you didn't do much; you just changed your diet and you just changed how you were working out. Well, that makes complete sense, because we see that, especially for people that are just starting their journey in longevity, making those simple changes has a huge impact. And so in that study, we showed that a lot of individuals who adopted a vegan diet for about 8 weeks were able to reduce those epigenetic clocks very frequently.
I think
As of
it's
last week, oh, sorry, go ahead. Go ahead.
no, go on.
Yeah, so as of last week, we actually finished up a paper which utilized ketamine treatment, and for that, 2 to 3 weeks, omic age was significantly reduced in those individuals that were undergoing ketamine treatment. So, we're starting to see that anywhere from 8 weeks to 2 to 3 weeks, depending on the type of intervention, you can start to make changes pretty rapidly.
It's really important as well, because one thing people often say to me is that this area, this field of aging and longevity is for the wealthy. And I have had a number of people commenting on the podcast saying, well, this is great if you've got tons of money, but, it's not for the average person. And actually, it's one of the reasons why the first things I wanted to do was, really just the basics. So, we're not looking at expensive supplements or anything like that. Really just diet, nutrition, exercise, movement, sleep, or all of that. And so, yeah, just wanted to bring that across, that it is really accessible.
Yeah, no, I agree, and I think that, I mean, quite honestly, I know that there's been a name that's being pushed out around, like, his name is Brian Johnson, who's spending, $1 million or however much money, and and I think it's great that he has the financial freedom to be able to do that. But yeah, like maybe somebody like me or somebody like the average listener who wants to be able to understand how much that they can reduce. That was the main thesis for bringing something like this to market, was that you'd be surprised at how many things that your grandmother, like especially my grandmother, has told me. Like in Indian food, like, a lot of it is turmeric based. There's a lot of good healthy fats that are being consumed. And if you listen to just the general public, fats are bad, you have to be on an extensive, like, high intensity workout or whatever. You'd be surprised at how much even a little bit can go a long way. And so I think that that's where this test really, that was the purpose of this test, is to give you objective values so that you can really understand what works for you, because something that works for you may not work for the other person.
That's a really good point, because one of the things I really found very valuable was that there's a lot of very actionable information in here. It's not just these are your ages, and it's not just something. What am I trying to say here? What I'm trying to say is that every area that the report looked at, and it goes really in depth. There's pages and pages. Everything was highly actionable. In terms of foods, in terms of maybe some supplements and things that you can change. Um,
Yeah.
can I ask you, what would you say is the most effective intervention that you've seen?
Yeah, and I will say that is a very hard question to answer, because just like I said before, something that works for everyone may not necessarily work for you specifically. But even with that said, I think a lot of the interventions that seem to show significant changes from the peer-reviewed literature. So I will say that my focus will be coming more from the peer-reviewed literature. Has really got to do with certain supplementations that revolve around, for example, omega-3. There was a fantastic paper that came out recently that showed that omega-3 supplementation by itself has been able to reduce the epigenetic age. So we're also using epigenetic clocks as the marker to show reductions. Has been able to reduce those upwards of anywhere from 3 to 4 years. In epigenetic age within the time frame. I believe it was a year supplementation. I will have to double check that paper, but omega-3 supplementation has been very impactful. Um, Honestly, of a short-term vegan diet, we've been able to show significant reductions during that time. Um, the One of the things that came from that is that you have to be mindful of what other types of vitamins that you're going through. So, like vitamin B12. So, if it's a vegan diet mixed in with vitamin B12, I think that that would make a pretty good reduction with the with those epigenetic clocks. We also show, and this is something that was interesting, is positive social interactions. Now, it's hard to quantify what a positive social interaction is, but essentially, if you are able to spend time with loved ones, if you're able to spend time with individuals that bring you literally like that emotion of happiness and joy, and that make you feel included into a collective, we've actually been able to see significant reductions in individuals that reported a positive social interaction. There was a brilliant study that was recently published as of, I think, about a month ago, where they were able to quantify the social interaction based on a particular score. And they showed that positive interactions, that as that score increased for that positive interaction, the epigenetic clocks, which include the different biological based epigenetic clocks, they started to decrease as well. So here it kind of goes to show that really the people that you surround yourself with have such a huge implication in the type of epigenetic age that you will be able to get out. And then also, I think other people have said this previously, but caloric restriction, if you do a 20% to 25% reduction in your estimated total calories that you typically take, you can actually start to see decreases in your epigenetic age as well. So, um, so I know I said like four of them when you asked me for one, but it's really to show that there's multiple ways of doing it. So whichever of these four that you haven't tried, pick one and just try to do it for an extended amount of time.
The social interaction one, I think, is so interesting.
Yeah,
We also run a dementia clinic, and I mean, there's very well documented research out there that social isolation is a big driver for the risk of dementia. And so, yeah, that's a really interesting one. And I think also um, when they looked at the blue zones, I know originally, people were going out there trying to find a certain compound. What is it that the people in this region are drinking or eating? They're looking for something in the soil or some kind of
yeah,
compound. And I think more and more, what we have begun to see is that in those regions, often there's a very strong social structure in those parts of the world as well.
yeah.
So that's really, really interesting.
It really is, and I mean, from a personal note, like coming from a society where traditionally or originally, mental health wasn't seen as a very important, discussed topic. I think that going through some of the literature and then also seeing how these markers have been changing with just something as in quotes simple, which I don't think it's that simple, but as simple as positive social interaction and also just healthier mind states, it's really got me thinking a little bit more about what the importance of just good mental health is and how that can relate back to your general biology as well. That's as deep as your epigenetics, deep as your molecular biology, if you will.
I love that. I really love that. And I mean, I've been in practice for 20 years, and I do think it is one of the things I, without a doubt, see is the impact on people's health when, there is emotional disconnect or social disconnect is huge. So that, that mental health and emotional health piece is absolutely critical in any kind of healing process.
Yeah, absolutely.
Varun, are there things you do every day? So, no, with the work that you do, it must have inspired you to maybe change your lifestyle and habits and things as well. Are there things you do every day?
Yeah, yeah, absolutely. And it's actually funny and very ironic because when I started at TruDiagnostic, a huge shout out first of all to the founders, Ryan Smith and Hannah Wendt, because when I joined, it was a very small burgeoning company, and so when I joined, it was right at the heels of my PhD, and I actually took the test, within the first like I think 6 months, and I was 7 years over my chronological age at the time, and which was kind of crazy because I thought I was eating well. I'm vegetarian. And also I was working out extensively. At the time I was doing a lot of powerlifting, like, deadlifts, squats, bench press, all of that. And so I didn't expect that result. And so what I've done to kind of change it is not only based on, tracking my own true health or true age and true health, uh, but also based on the papers that were coming out, I started employing caloric restriction at least during the work days. I like food, so the weekends I'll be a little bit more liberal with what I consume. But at least during the week, I try to employ 25% reduction. So for me, it looks about 1400 kilocalories or so. maybe less depending on, or more depending on what I work out. I've changed my workout routine to be a little bit more cardiovascular based, because not only from the epigenetic side, but a lot of the literature that's been coming out in longevity is, cardio and building up your cardio has actually showed significant associations to increased mortality. So that's one thing I've changed, and that I think has contributed to my decrease in my epigenetic age. And then, and honestly, just trying to understand that there's more to the world than work. I maybe my my colleagues at TruDiagnostic won't like me saying that, but yeah, there's something more to this world than just working. I mean, don't get me wrong, I love what I do, and I think I try to find the beauty in being able to develop things like this, which satiates the creative side of myself. But I think knowing that there's a bigger part of the world that is about social experiences, experiencing different things around the world. So traveling. Anytime I go out for conferences, I try to make it a priority to at least see something, observe another culture, meet new people. And so those things I've definitely included, because traveling is very hard. Actually, I've noticed that even when I travel, my epigenetic age increases like crazy within a short amount of time. So as a way to kind of deflect that, maybe do something fun. So those are three things that I've started to implement in my routine.
Love it, love it. Yeah, and that work-life balance, isn't it? It's so important. Really, really
And
important.
unfortunately for Americans, yeah, for Americans, the work-life balance is not the best. I know a lot of the many of the European colleagues that I've had, they do a very good job of it. So just taking a few pages out of their book.
I can tell you the difference is huge between the US and Europe. I mean, it's crazy the work ethic in the US. So, um, yeah, my sister, who lives in Colorado,
Mm,
she has a virtual job, did not even tell her employers she was having a baby. So she just
wow.
took 2 days off. And in Europe we have very different.
Parental leave, yeah.
Yeah, but yeah, no, I know. So, but it's important to get that
Yeah.
work-life balance. Right. Um, what's the future? I'm kind of curious because I think, we see with AI and technology and all of the pace in which things are going, there's just the speeding up. What is the future of personalized testing and epigenetic testing?
Yeah, absolutely. This is a question that we always ask ourselves at True. Yeah, I think the main thing for us is we would love for more adoption of these epigenetic tests. I will say, like, even amongst the scientific community, epigenetic clocks are still highly contested because, some of the times people are asking, like, do we really know what modulates these epigenetic clocks? My personal opinion, and which is, probably founded on a lot of these trials that we've run, is I think we're starting to understand this better. We are running a lot of these clinical trials where we're starting to understand how, like, which interventions are reducing these clocks and what are those interventions really hitting, which is then giving us an understanding of these clocks themselves. So in the next 5 to 10 years, what we would love is for these to be commonplace, that everyone understands, everyone is taking these tests, because then they can understand how these are changing in the context of your regular lab panels. Um, we don't want to replace all of the current lab panels just yet. It would be very important to understand both of them together, but it would be great for the next 5 to 10 years. In terms of development, some of the things that we're looking at is how can we employ AI, like artificial intelligence, to be able to take all that information that we provide to you. I think I heard you say that you got a huge report, like, I don't know, 24 pages worth of information. How can we take that and develop AI tools that can actually convert that information and give you more actionable insight? So it'll, for example, something that we were working on at our company is, can we create almost like a chat GPT, which is able to take all this information along with your clinical data, your VO2 max data, your, let's say MRI data and BMI data, and be able to give you a comprehensive look of here's your epigenetic health and here's your output health or your phenotypic health, and this is what you need to do based on all of these measures. And so we're working on things to kind of take into consideration all of this information. So we're actually working on this. There's our entire engineering team, fantastic. They're really working on creating these type of not necessarily large language models, but these type of AI models to be able to develop that. And then last, actually, this is a project that I'm personally working on, but it's also very interesting, is companion diagnostics. So, for example, recently we published a paper that suggested that the usage of Ozempic or semaglutide can reduce your biological age. But what was interesting in that study is that we're showing that Ozempic has an impact on your epigenetics. So now can we create a predictor of whether Ozempic is right for you or not? A pharmacological drug, if you will. Are you actually right for that drug? You can use genetics to kind of figure that out, but what we're also seeing is that on the epigenetic side, we can track a lot of those changes as well. So these companion diagnostics are another thing that we're working on. So there are three things that we're really excited about in the near future, and what we're hoping for. We really just think that epigenetics, these epigenetic biomarkers, can be very, very useful in a whole span of things related to biological age and also human health in general.
Yeah, so I mean, obviously, there's with Olympic, what we are seeing is that some people do incredibly well with it, others end up
Yeah,
with complications, and it would be great to have that kind of information beforehand. You
yeah,
know how you're going to respond and whether it's the right thing for you. But also talking about bringing this more into clinical trials and things. I did recently speak to Professor Jan Manson, who's the um, head of preventative medicine at Harvard University, and they've just done the vital trial on telomere length and vitamin D.
yeah,
And so it's really nice to start to see research beginning to incorporate this in, because um, aging is still the number one risk factor for just about any disease, whether it's
yeah.
cardiovascular disease, dementia, cancer. Aging, as you age, your risk for all of these things go up. So actually, what you guys are doing, it's not a luxury. And very often people think, oh, well, this epigenetic testing and things, it's a luxury if you've got a bit of extra cash lying around. I really would flip that and say that if we can modify aging, if we can influence aging, we can influence so many disease processes out there. So it
Right.
genuinely isn't a luxury. It should be at the core of clinical practice.
Yeah, and one of the things that we're really trying to do is we understand that like finances are an important thing to consider, and so a lot of the work that we're doing now also is we're just trying to figure out ways to bring down either A, the price, or B, with these clinical trials, have the FDA approval, because a lot of these different types of tests are not FDA approved, not because that they're not important, it's just because we don't know much about them yet. And so, with that said, a lot of what we're doing now is once we can get FDA approval, then hopefully insurance companies will be able to reimburse this. And so we're working on this from the corporate side as well, trying to bring this to the masses.
So I'm also very excited to say that Regeneris Laboratories here in the UK, who are your partners in bringing the test and making it available and accessible in the UK, is offering our listeners a 15% discount
Wow.
on the True Age and True Health testing. We haven't spoken about True Health today, but which is a whole new big thing. But
Yeah.
there's a 15% discount for our listeners. And all they need to do is just email in to Charmaine at charmaineshepherd.com and ask for the discount. We can get that applied for them. It's been a great honor to have you on the show. And I must say, the conversation took some turns which I hadn't expected, around that kind of social and emotional elements. And I absolutely love that. I think it's such an important thing to look at. And I think just really brings it home how, there isn't just one or two things that impact on this. It's multifactorial. There's so many things that that play into this. But I think the key message is there's so much that people can do to really influence the rate at which they age.
Yeah, the solutions are really at your fingertips. So you just have to know what to look for. And I think that that's where we can come in and help.
Well, listen, thank you so much for your time. I really, really appreciate having you here with us today. And I hope you enjoy the rest of your day. Take care.